So the primary somatosensory area we're going to come back to in just a minute, but that's where those somatic sensations are going. That area in blue, we're going to be talking more about that in just a second. Let's see here. So the primary somatosensory area is located in the post-central gyrus, the parietal lobe of the cerebral cortex, and that's, again, this is a lateral view. This is the anterior side, so this is the frontal lobe right here of your frontal bone. There is a groove called the central sulcus that separates the frontal from the parietal lobe, and then the temporal lobe would be below that. That would be, like, parietal would be here, temporal on the sides, occipital back here, that's the occipital lobe there. But in terms of somatic general sensations coming from touch, pressure, pain, temperature receptors, vibration, itching receptors, all those proprioceptors, all that information is going to this blue area. This is a ridge of the brain. You have grooves called sulci, and you have ridges called gyri. The post-central gyrus is posterior to the central sulcus, hence the name post-central gyrus. There's also a nearby somatosensory area in the parietal lobe that is involved in memory, so that you remember sensations that you're feeling, from these sensory receptors. And this has been mapped out. So this is called the sensory homunculus, and this is a frontal section. If you look at the picture up here, this is taking a section this way and then turning it around. So you're seeing the cerebral cortex, which is the gray matter. The white matter is underneath, and that's what is transmitting information between different parts of the brain. This is the longitudinal fissure. That's that deep fissure between the hemispheres. So this is showing you the post-central gyrus in a frontal section, and this would be on one side of the brain, so this would be, I guess, the right cerebral hemisphere, but it'd be the same on the left. And because of studies such as taking somebody who's been having brain surgery, a volunteer with brain surgery, and stimulating with a electrical stimulus different parts that gyrus, you could they would feel sensations in these different parts of the body. So if you stimulated this area, the person feels tickling or something in the toes. So they mapped it out, and what they've done is they've drawn what's called the sensory homunculus. A homunculus is like a miniature human. It's been used in a lot of different ways, but in this case, what it is it's a mapped out version of all the different parts of the body, and the size reflects the amount of sensitivity. In other words, the density of sensory receptors, which also means if you have more density of sensory receptors, you have smaller fields, smaller fields in which you're feeling those sensations. So, so for instance, the hands and face have the most sensitivity. They've got the most density of sensory receptors, therefore they have the smallest receptive fields and the largest density of sensory receptors. And whereas the legs and trunk, this is supposed to be the trunk right here, that's the rear end, and that's the trunk, and that's the arms. Arms, trunk, legs, not as sensitive. So the homunculus just refers to this kind of like little weird looking person or human form. Usually a homunculus is kind of, has exaggerated features, and that's why they call it the homunculus in this case. Any questions about that? So this just shows that certain parts of the body are much more sensitive than others. There's also mapping of the motor areas. The motor areas of the brain have also been mapped out, and the motor areas of the brain are found in the frontal lobe of the cerebral cortex. The primary motor area specifically is the precentral gyrus, right in front of the postcentral gyrus and in front of the, or anterior to the central sulcus. And this area, the primary motor area, is involved in controlling skeletal muscle contractions. And again, when you look at the, well, let's look at that area, and then we'll look at the homunculus. This is the frontal lobe, and, or this right here from the central sulcus forward is the frontal lobe. The red gyrus is the precentral, meaning anterior to the central sulcus, precentral gyrus, and it's been mapped out as we'll see. That controls general muscle contraction, just, you know, moving around, doing things that you generally do. But there's also a premotor area in the frontal lobe, and that premotor area right about here controls learned movements. So, you know, unlearned movements that you just generally do, you just do instinctively, is controlled by primary. Premotor area is also involved as well when you're doing something learned. So you'd be using both, but if you're learning to, you know, type or play a musical instrument or do anything like that, that requires a little bit more control, and the premotor area is involved in that. There's also, ignore the gustatory area, that's a sensory area, but the frontal eye field area, as the name implies, controls eyeball movement. So when you're moving your eyes around, that's controlled by this frontal eye field area, which is area 8. And then there's the Broca's speech area. This area here, 44 and 45, contains the Broca's speech area, which it controls muscle movements in your mouth and throat, and, you know, the skeletal muscle of your mouth and throat involved in speech. So all of these areas, these motor areas, are all in the frontal lobe, and all are involved in controlling skeletal muscle contraction. So here's this is specifically the precentral gyrus right here. So if you look, you always want to look at these little pictures up here. This is the precentral gyrus in the frontal lobe, and a frontal section that they now are allowing you to see from an anterior view. There again is the longitudinal fissure between the cerebral hemisphere. So this is one hemisphere we're looking at, the right hemisphere. And it looks very similar to the sensory homunculus, because just like the areas that are the most sensitive also have the most precise types of movement, and therefore have the smallest motor units. And by that I mean the number of muscle cells that are stimulated by a single motor neuron. So let me go back to this, I'll come back to this picture. So remember, a motor unit is a motor neuron and all the muscle cells it stimulates. So these are somatic motor neurons in this case, as opposed to the autonomic system. And so the purple neuron stimulates these two cells. The green neuron stimulates these three over here, the darker ones. So let's say these are white fibers and these are red fibers, you know, that you have in your muscle. These are stimulating specific muscle fibers. And motor units can vary in size. If it's a small motor unit with just a few muscle cells per motor unit, it's going to be involved in very precise contractions, like, you know, contractions of facial expression, movement of your eyes, and just, you know, overall movement of your face, speech movements, as well as movements of your fingers. So your face and your hands are where you have the most precise contraction, and you have the smallest number of muscle cells per motor unit. So when I say a small motor unit, it means the number of cells that are stimulated, there's a small number. It might be just 2 or 3 or 4 or a dozen, but it's going to be a small number. If it's a muscle in your shoulder or arms or trunk or legs where you don't need precise contraction, you're going to have hundreds or even thousands of muscle cells per motor unit. In other words, a single muscle cell doesn't just stimulate 2 or 3, or a single motor neuron, I should say. A single motor neuron doesn't just stimulate 2 or 3 muscle cells. It stimulates hundreds or thousands. It might be a couple thousand that it stimulates. Because in your trunk and arms and legs, you don't need precise control, you need strength. So where you need strength, you have large motor units with lots of muscle cells. And that's reflected in the motor homunculus, in the precentral gyrus. Your hands and face have the most precise control and have the most innervation and the most area of the brain devoted to that innervation. So you have more areas and larger areas devoted to facial expression and speech and eye movements. and precise contractions of your hands, and less precise control of your trunk, arms, and legs. Okay, so the two homunculuses look pretty similar to each other. Any questions about that? Now, we're going to just talk very briefly about the motor pathways, because this chapter deals not only with sensory, general sensations or somatic sensations, but it also deals with a little bit about the brain control and motor pathways. So the somatic motor pathway is first the upper motor neurons that are stimulated in the cerebral cortex. They're being stimulated by interneurons in the gray matter of the cerebrum and the cerebral cortex. They in turn stimulate lower motor neurons, which in turn stimulate skeletal muscle to contract. So this picture, which I've shown you over and over again, shows you the somatic motor pathway. Here is the upper motor neuron. The cell body and dendrite, this is multipolar, so the cell body and dendrites from the gray matter being stimulated by some interneurons in the gray matter. When it's stimulated, it's going to generate action potentials that run down from the brain through the brainstem, crossing over in the medulla oblongata, and crossing over to the other side of the spinal cord, and then synapsing with the lower motor neuron. It's hard to read that, but on your computer screen or book, you can read that. The lower motor neuron is, there's a synapse here between the synaptic end bulbs of the upper motor neuron and the cell body and dendrites. So the lower motor neuron, again, multipolar neurons here. The axon of that lower motor neuron exits the spinal cord through the anterior nerve root. Remember, anterior is where motor impulses travel, posterior is where sensory impulses travel. Out to the muscle, and in this case, maybe stimulating muscle contraction of the hand. Yeah?
Can I shut the door?
Oh, yeah, I'll do that.
Okay, thank you. I was getting distracted.
Yes. You should try to close the door instead of left it open a little too long. Okay, so notice that in his case, the left hemisphere is controlling the movement of his right hand, and vice versa. The right side would control the left side. Also, it's showing him contracting muscle in his hand. If it's contractions that just are involved in maybe pinching the pen, just generally pinching the pen, that would probably just be the primary motor area. But if it's muscle contractions involved in moving the pen and writing, then that would also involve information coming from the premotor area, which is the one that controls learned movements. But again, you have crossing over. This crossing over happens either in the medulla oblongata, but it can also happen in the spinal cord, just like with sensory decussation. Decussation, by the way, that word means crossing over. So when you see decussation or decussates, that means it's crossing over, either crossing over, from sensory up or from motor down, but it's crossing over. So the sensory motor functions in the somatic system are crossing over. And this picture, the corticospinal pathway, simply means from cortex to spine, corticospinal. And so you either have, here's the upper motor neuron coming down through the midbrain, down to the brainstem, into the, eventually into the medulla. This is the medulla oblongata, and there are tracks, motor tracks in the medulla called the pyramids. You may have learned about the pyramids in the brain. They are located in the medulla oblongata, and these are motor tracks where you have crossing over or decussation happening, and that's where you have that motor information crossing over, in this case from the right side to the left. And then that lower motor, or that upper motor neuron synapses with the lower motor neuron here that exits out through the anterior nerve root out to skeletal muscles in the distal parts of limbs. I'm not concerned about where the impulses are going. I just want you to know that depending on where they're going, like here they're going to the to other parts of the body, like the trunk and the proximal parts of limbs. I'm not going to ask you questions about which one is, which one crosses over, in this case or this case, but you should just know that depending on where that impulse is going, depending on the destination of that impulse, the muscles that are going to be stimulated, you either have crossing over or decussation in the medulla oblongata of the brain, which is in the brainstem, or you have decussation in the spinal cord. So here the upper motor neuron crosses over from right to left, synapses with the lower motor neuron. So there's crossing over in the spinal cord or the medulla in those motor pathways. Any questions about that? Which fortunately, it's the same as sensory, so it kind of makes it easier to remember. Now, you also have regulation of the upper motor neurons, not just stimulation to cause movement, you also have regulation of the upper motor neurons. And this involves the basal ganglia and the cerebellum, which regulate the activity of those upper motor neurons coming from the cerebrum. The basal ganglia, I'm going to show you a picture of them. These are deep inside the cerebrum. They function in starting and stopping movements and in preventing unwanted movements. You may know that Parkinson's disease usually affects the basal ganglia. And as a result of the basal ganglia not functioning properly, you might get unwanted movements, jerky movements, or tremors. And those are symptoms of Parkinson's. That indicates what the basal ganglia does. It prevents those unwanted movements and jerky movements and tremors from happening. And the basal ganglia is in the cerebrum, deep inside the cerebrum. You can't see it in this picture or this one, but it is deep inside. This is all, what you're seeing here is gray matter. Because the gray matter, the cortex extends even into the longitudinal fissure. Remember the hemisphere, or the fissure that separates the two hemispheres? The gray matter and gyri and sulci all extend deep into the longitudinal fissure. The only way to see the basal ganglia, because this is gray matter, is to go deeper into the brain. And this picture is kind of supposed to be three-dimensional, but the blue areas here, these blue areas that are on both, in both hemispheres, those blue areas are the basal ganglia, and these are clusters of neuron cell bodies. They're gray matter. The red is the thalamus, and as you can see in this frontal section, if you're looking at the frontal section of the brain, these are the two cerebral hemispheres over here. That's the cerebral cortex of gray matter right there. That's the white matter. The corpus callosum is the tract that connects the two hemispheres. There's a big ventricle here full of cerebrospinal fluid. And then the deepest part here, the thalamus and hypothalamus, the red and the light blue, that's the diencephalon region. That's not part of the cerebrum, that's a separate region. But the blue areas are actually considered to be part of the cerebrum. They are gray matter. They're clusters of neuron cell bodies. They look like this when you cut that in a frontal section. But these clusters of neuron cell bodies are called the basal ganglia, and the different parts have different names. You don't have to worry about the different names. But the term ganglia is a sort of a misnomer because a cluster of neuron cell bodies in the central nervous system is a nucleus. And some of these do have the name nucleus in them. Sometimes people do call the basal ganglia basal nuclei. That would be the more correct term, because a ganglion is a cluster of neuron cell bodies in the peripheral nervous system, like the posterior root ganglion and the autonomic ganglia. But its name has been used for so long, it's just the name most people use. But you do see sometimes it referred in books or by people as the basal nuclei. It's the same thing. And this is regulating upper motor neuron contractions or upper motor neuron stimulation of muscle to prevent unwanted movements, prevent tremors, and things like that. Now, the cerebellum, any questions about basal ganglia? On this picture, you can see the thalamus, but the basal ganglia would be, you can't see in that picture. Anyway, the cerebellum, we're going to talk about, that's the cerebellum there and there. The cerebellum also has gray matter on its surface, and it has white matter that looks like a little tree on its side. That's called the arbor vitae. That's the tree of life, because it looks like a tree. And it contains white matter that allows for transmission of impulses between other parts of the brain and the cerebellum. And what the cerebellum does is it coordinates muscle contractions and controls posture and balance. We're going to look at that in a little bit more detail. So one of the things that the cerebellum does is it kind of regulates and corrects muscle control. So it's monitoring skeletal muscle movements because it monitors proprioceptors. So when you're moving your arms around and you're getting proprioception information, that information doesn't just go to your cerebrum, it goes to your cerebellum. And so your cerebellum knows how you're moving, it's monitoring that, and it makes corrections as necessary so you're not flailing around wildly. So it helps in coordinating muscle movements by regulating and performing corrective feedback to the upper motor neurons. It's also active when you're learning movements, when you're learning skilled movements like throwing a baseball or typing or playing a musical instrument or anything like that. And then it would also include speech movements because you have to learn how to talk. And a child learning to talk is, you're going to have very precise control of that. The cerebellum is active in that regard as well. And the cerebellum, as we'll see in the next picture, it receives input coming from the eyes, from proprioceptors throughout the body, and from equilibrium receptors in your inner ear that we'll be talking about later. And it's monitoring all that information that's coming in about your body position and your balance, and it makes corrections as necessary to maintain your posture and balance so you're not leaning, to one side. When a person drinks alcohol, one of the places, that's going to be suppressed in the brain, its activity is suppressed, is the cerebellum. And so you kind of get an idea of what the cerebellum does when you look at somebody who's drunk. They can't walk straight, they're slurring their words, they can't properly speak, they're not able to coordinate the contractions they need for speech or for walking and keeping their balance, and touching their nose, things like that. So you can get an idea of what the cerebellum does by simply watching what happens in a person who's really drunk. And all of that is, this firing of muscle movement is due to the lack of cerebellum activity. This picture is showing all this feedback. It's kind of hard to follow. And then, unfortunately, this is blue here. I'm going to have to get a different color for this background. I'm not sure why it's so dark blue, but I'll see if I can fix that. But anyway, here is the cerebellum. And the cerebellum cortex is what's doing all this function of monitoring sensory information and then controlling corrective output of muscle. So you've got all this information coming in to the cerebellum, information coming from the cerebrum. The cerebrum not only is sending signals down to control muscle movement, but it's sending signals to the cerebellum so the cerebellum can make corrective feedback as necessary back to the cerebral cortex to keep the muscle coordination going. This is the thalamus. The thalamus does relay that information between the cerebellum and the cerebral cortex. So it's another relay station there, once again. The cerebellum, as it says here, receives sensory signals from proprioceptors in your muscles and joints. The vestibular apparatus, that's the equilibrium receptors in your inner ear. And then visual information coming from your eyes. And all of this informs the cerebellum about your body position and whether it needs to correct your body position and posture so that you're standing straight up and not leaning or falling over. So it's making corrections as necessary. So it helps with corrections involving coordination of muscle contraction and corrections that help you maintain your posture and balance. I think that's the last picture in this chapter. Any questions before we move on? Okay, so I think that was the last one there. So let's move on to the special senses. These are the senses that are found in your head, and this is in the next chapter, which is, what is that chapter number? Chapter 18, 17, okay. So the general senses versus the special senses. General senses include the somatic sensations, of tactile sensations like touch and pressure and itching and vibration. Those are tactile. Thermal, which is heat or cold, pain, and proprioception. These are all considered to be somatic sensations. That's what they're called, somatic sensations. And then visceral sensations, which are sensations coming from your viscera, and some sensations you feel like pain and discomfort, but others and stretching of your bladder, you feel that. But many visceral sensations you are not consciously aware of. You're not consciously aware of your blood pressure and your pH of your blood. Those are visceral sensations. And these are found throughout the body, and they consist of simple sensory structures, either sensory neurons or receptor cells that stimulate sensory neurons, but they're not very complex. They're just basically sensory neurons or the receptor cells that communicate with the sensory neurons. On the other hand, special senses, they are the senses of smell, taste, vision, hearing, and equilibrium or balance, and they are concentrated in your head. And the reason why it's concentrated in your head is because when animals first evolved and they started first evolving these senses, these animals were swimming in the ocean and swimming, if you're swimming towards something, the first thing that's going to encounter, if you have a head, the first thing that's going to encounter various things like predators and prey and other things and hiding places and all that, that first thing that's going to encounter all that is your head. So it makes sense that during evolution, the most logical place that you would evolve eyes and ears and smell receptors and all that would be in the head region, which is the first part of the body that enters an area, if you're swimming like a fish or, other organisms that, the fish, but other, trilobites. If you look at a trilobite, they've got their sensory receptors in their head. So very early animals, if you get beyond like things like jellyfish and get beyond the sponges and things like that, we get into those organisms that have bilateral symmetry like we have. Bilateral symmetry meaning right and left sides, and having a superior and inferior and anterior posterior parts of the body. Those organisms very quickly evolved their senses in their head. Of course, we're upright, we're not moving into the area with our head, we're moving into the body, into the region upright, but still, it's helpful to have all these senses in the head region at the top of the body. Now, these structures are anatomically complex. They're not just simple sensory receptors. They have sometimes complex neural pathways. They also have low threshold for stimulation. It doesn't take much to stimulate these sensory receptors in these special senses. We're going to start with olfaction, the special sense of smell. And we're going to be looking at a picture of this, but the olfactory epithelium is what they call the region, the olfactory region in the upper part of your nose, the mucous membrane, just below the cribriform plate of the ethmoid. I'm going to show you pictures of this so you'll hopefully remember the terminology. But just below the cribriform plate, you have these regions called olfactory epithelium. It's kind of a misnomer because it's not really epithelial tissue, it's nerve tissue and other cells, but it's in the mucous membrane, which is made of epithelium and connective tissue, so they call it the olfactory epithelium. There are, in the olfactory epithelium, there's 10 to 100 million olfactory receptors. These are first order sensory neurons. They're bipolar neurons, they're not unipolar. 10 to 100 million, and these guys detect 10,000 different odors, what we call odorants, or odorant chemicals. So you see the term odorant, that means a chemical that stimulates one of these receptors. This is very detailed. 10,000 is a lot. This is more than what your taste receptors could detect. So you have very specific receptors that detect all these different smells, and these are the receptors are part of the sensory neuron. But they call these cells olfactory receptors or olfactory receptor cells, but they're not really receptor cells like I've been defining. It's an unfortunate term because of that. These are really sensory neurons. Their dendrites contain the sensory receptors. It's not a separate receptor cell and all that. And I'll be showing you that in a second. There's also in the olfactory epithelium, there's supporting cells that protect the receptor cells, that nourish them, and also insulate them, electrically insulate them, kind of like the myelin sheath insulates. But they're surrounded by these supporting cells that support them. And then there's also basal cells, which are stem cells that divide and replace those olfactory receptor cells. Oddly enough, we've talked about before, or I'm sure whoever your instructor was that you had last semester told you that neurons don't get replaced. They die, and if they die, they're dead, they're not going to get replaced. However, there are certain sensory neurons that can die and do die and are replaced. And that would include the olfactory receptor sensory neurons. They only last for about a month or so, possibly because they're exposed to a lot of chemicals. They're right there at the body, at the inside of the inside of the mucous membrane. They're right there at the surface, exposed to various chemicals, and probably it's kind of hard on them, but anyway, they don't live for very long, just for about a month. And so the basal cells divide and replace them. So here's a picture of a sagittal section of the face and cranium. Here's the forehead, there's the brain, and here is the cribriform plate, and just below the cribriform plate, which is right here, below that is the nose. Your nose is just underneath this, and that purple area is the olfactory epithelium or olfactory region. The rest of this is just plain mucous membrane in the inside of your nose. So your olfactory region is right under the cranium. I've got a picture of that here. Here's the cranial floor. This is what we're talking about. The cribriform plate is part of the ethmoid bone, and all these little holes are these holes that you see right here where the sensory nerve fibers are passing through. Those teeny tiny holes are called olfactory foramina. Foramina are tiny holes. And olfactory meaning the olfactory sense of smell. It's not the smell itself that's going through there, it's those sensory fibers or axons that run through that on their way to the brain. So what we just saw, that cribriform plate is right here and right here in this enlarged picture, and this is the olfactory foramina, the little holes. And they only look about the size of maybe if you took a pin and you poked holes in the bone. They're not very big holes. Now, below that is the olfactory epithelium. You've got the epithelial layer, which is really more like nerve tissue, but it's got, epithelial cells and neurons in there. There's connective tissue, so together that makes the mucous membrane, but this mucous membrane contains receptor cells. The cells in blue are the sensory neurons. They're the receptor cells. They are bipolar. I'll show you a picture of bipolar in a moment. These cells have little hairs. They're called olfactory hairs, but they're not really hairs. They are actually what are called cilia, but they're not the kind of cilia that wave. You do have cilia that wave inside the nose and wave mucus to the back of your nose. These olfactory hair or cilia are actually not waving or moving, but they're called cilia because they are like little hairs. They just function differently. So they're not moving, but they are embedded in the mucus layer, or they call serous secretion. It's really a mixture of mucus as well as tear secretions that come from your eyes. And so you've got this mucus layer that the hairs are embedded in. And those olfactory hairs are what gets stimulated by odorant molecules. So the odorant molecules are the little dots here. It could be anything, fried chicken smells or cigarette smell, cigarette chemicals, things like that you can smell. And that tickles those hairs and stimulates those hairs. And we'll see how they're stimulated, but that stimulation causes a depolarizing generator potential to be generated, and that then results, that generator potential is going to run up the dendrite, this is the dendrite, and down or up the cell body to the beginning of the axon, to the trigger zone or axon hillock. And that's where action potentials get generated and then propagated up through this axon, up through the cribriform plate. And we'll talk more about that in just a minute, but we'll come back to what happens up there. But those axons are running up through the cribriform plate. Now there are also other structures here. For instance, the supporting cells are shown in brown. These are the ones that protect and insulate the neurons. The basal cells are at the kind of the base of the epithelium. These are the cells that divide and replace the dead receptor cells when they die. Keep in mind that our olfactory region is very small relative to other animals. Primates in general have a smaller sensory area than other animals do because we don't rely as much on our sense of smell. We rely more on our vision, to help us get around. So dogs, for example, especially if they have a big snout, they have a really large olfactory region, especially like bloodhounds that are bred for that function of smelling things. But dogs and other animals have much bigger sensory areas and much more sensitivity to smell. You also have these olfactory glands that produce mucus. So there's mucus production happening here to keep everything nice and moist. In order to smell anything, you have to have mucus. You have to have a mucus layer. If it were perfectly dry inside your nose, then you're not going to be able to dissolve those chemicals in the mucus, and you're not going to be able to stimulate the receptors, and therefore no smell sensation. So you have to have that moisture in your nose, and you're getting that moisture from those olfactory glands. You're getting moisture from the tears coming into your nose through the nasolacrimal duct, as we'll see later. You're getting mucus from goblet cells, which are scattered throughout the epithelium. So there's a lot of mucus production going on in your nose. There's also mucus coming from your sinuses. If you remember the paranasal sinuses in your forehead, in your cheeks, in your ethmoid and sphenoid bone, they're also draining mucus. So there's a lot of mucus production, a lot of mucus going into your nose. Okay, now this, I just want to show you, this is a bipolar sensory neuron, but it's in a reverse direction. The dendrites are pointing up, and so this is where these would be olfactory hairs or cilia. They wouldn't look like this exactly, but they're more like cilia looking, and I'll show you a picture of that in a minute. And so when the cilia get stimulated, then you get a generator potential that travels down to the trigger zone, and if you reach threshold, then you're going to get an action potential. And again, the threshold for stimulation is lower in the special senses compared to somatic senses, and so it doesn't take much to get that action potential stimulated. And then from there, this is what runs through the cribriform plate up to the olfactory bulb that we're going to look at in a moment. So again, the cilia of the dendrites are pointing down. That's the dendrite, and then that's the axon. Now the axons, all these axons together are considered to be the olfactory nerve, or nerve number 1. It's kind of weird because they're not one nerve or a pair of nerves. They are a bunch of nerve fibers, kind of, clustered together but still separate. But as a collection, they are considered to be the olfactory nerves coming from each side of your nose. Because you've got an olfactory region here and an olfactory region here. They're going up into the up through the cribriform plate. So collectively, all of those are considered to be olfactory nerve 1, and those sensory neurons then synapse with the dendrites of a second order neuron up here. This olfactory bulb neuron is a second order neuron. These are first order neurons. The ones that are stimulated are first order. They synapse with second order olfactory bulb neurons in the olfactory bulbs, which are enlarged bulbs above the cribriform plate. These are actually masses of gray matter. They're kind of like ganglia. They're very similar to ganglia, but they're not called ganglia. They're called the olfactory bulbs. But they're made of gray matter because they're full of cell bodies, and so a lot of not so much myelin sheath in there. Let me see what else I'm going to tell you here. Okay, so Generator potentials are produced in those olfactory hairs or cilia in response to chemical stimulation, and we'll see how that works, by odorant molecules. Remember, the term transduction means the conversion of a stimulus to a generator potential in this case. And these are depolarizing potentials. You know, these are graded potentials or depolarizing. If the generator potential it reaches threshold, it stimulates an action potential or nerve impulse in the axon, and that nerve impulse is propagated up through the cribriform plate, through the olfactory nerve, to the olfactory bulb, where you have synapses. And somewhere in here I had it. Okay, well here's, before we go to that, let's look at this picture. This is actually showing an electron microscope image magnified 5,000 times with an electron microscope. The blue cell, it's colorized blue. This is the sensory cell. This is the olfactory receptor cell, the sensory neuron, first order sensory neuron. Here's the olfactory hairs or cilia. And so when they get stimulated by an odorant, they generate a generator potential that travels up to the cell body, and then at the axon it becomes an action potential. And there's supporting cells, the brown cells around there are supporting cells that are protecting those cells. Now, the physiology of olfaction, I'm going to go through this in a picture format, but it's all written out for you here. So let's look at the picture. And the picture I'm going to show you, which is basically this one here, just been enlarged, is happening in the plasma membrane and cytoplasm of the olfactory cilia. So the blue is outside. This is extracellular fluid, which would be mucus. This is the phospholipid bilayer. This is the cytoplasm. So embedded, again, in those olfactory cilia, those little olfactory hairs that you can see in this picture, embedded in those olfactory hairs or cilia are olfactory receptor proteins. And they bind to specific odorant molecules. If you can smell it, you have a receptor for that odorant. When an odorant molecule binds to the receptor protein, this in turn activates something called a G protein. And I'm going to actually go to the next picture because it's bigger. Okay, so here's an unbound receptor protein. Here's a molecule binding to the receptor protein. That's the extracellular fluid, which is mucus. Okay. So the odorant is dissolved in the mucus. When the odorant binds to the receptor, this activates a protein called G protein. G protein is found in a lot of cells of the body. It's used as a second messenger system. We call these second messenger systems because like a first messenger would be like in this case, an odorant molecule, but a first messenger could be something like a neurotransmitter or a hormone or something like that. Second messenger systems happen inside the cell, and they're a way of getting things to happen. It's how you kind of create a cascade of events that leads to an outcome. So odorant molecule activates G protein. We'll see that G protein activation is common in other sensory neurons as well. G protein activates adenylate cyclase, another protein. It's an enzyme. It's embedded in the plasma membrane of those cilia. And what adenylate cyclase does when it's activated by G protein, it breaks down ATP into cyclic AMP. Cyclic AMP is adenosine monophosphate. And cyclic AMP then activates this sodium channel. Now this sodium channel is closed unless it gets activated. It's a protein channel. It's a gated channel. It is activated by cyclic AMP. And when it's opened, and by the way, this of course is going to release energy, so that's part of the process, is you're energizing that protein, which allows it to open. The sodium channel opens, in comes sodium, and remember sodium's positive, so guess what? It depolarizes the cell, generating a generator potential. The generator potential travels down the dendrite or up the dendrite to the axon hillock, where it then generates an action potential. This is what transduction is right here, the transduction of a stimulus into a generator potential. That's transduction. That's what the word transduction is referring to, is this process, in this case anyway. So again, odorant molecule binds to the protein. This activates G protein, which in turn activates adenylate cyclase to break down ATP into cyclic AMP. This activates the sodium channel, it energizes it, and activates it so it opens. Sodium comes in, generating a depolarizing generator potential. And that generator potential, which is being triggered here, this is happening in these little hairs. That generator potential travels up the membrane to the trigger zone. There it triggers an action potential. The action potential arrives here and triggers the release of neurotransmitter that then binds to these dendrites. Now these are kind of weird cells. They're a type of interneuron in the olfactory bulb, and they're called olfactory bulb neurons, and they're second order neurons. They're inner neurons. They have a kind of weird shape to them, kind of different from other multipolar neurons. But these things here are dendrites. And so there's synapsing with those dendrites, and that neurotransmitter stimulates a depolarizing graded potential or postsynaptic potential. You can say that excitatory postsynaptic potential in this neuron, which in turn generates action potentials down the axons that run down the olfactory tract. So what's happening is you're sending a signal here up, and then you're synapsing here, and then these guys are sending a signal down the olfactory tract, and the olfactory tract carries that signal to the brain. You may remember, hopefully you learned about, this is just telling you what I've already told you, but as it says here, the olfactory bulb is considered to be gray matter. Even though it's not inside the brain, oddly enough, it's outside the brain, it's still considered to be part of the brain, kind of a little external extension of the brain. And that gray matter bulb contains those second order olfactory neurons, inner neurons, olfactory bulb neurons, which are inner neurons, and those then transmit information to the brain through the olfactory tract. So remember, so this is above, this is an inferior view of the brain, this is above the cribriform plate, okay? This is the olfactory bulb, it's full of those olfactory bulb neurons. This is where the synapse takes place between the olfactory receptor cells and those second order neurons. Those neurons in turn send signals down their axons through this tract. So this is not an olfactory nerve, this is the olfactory tract. Even though it's actually kind of external to the brain, it's considered an extension of the brain. Think of this as an extension of the brain. These others are called nerves. That's a tract. It's a little weird, but that's the case. So anyway, the olfactory tract continues into the brain, and from there, so here's what we just looked at. There's the bulb. The bulb transmits that information into the brain, and it goes directly to the primary olfactory area of the cerebral cortex. Notice it bypasses the thalamus. Olfaction is the only sense that does not get relayed through the thalamus. It just goes right straight to the primary olfactory area. This is in the temporal lobe. It's in the kind of deep, this gray matter kind of goes a little bit deep here. See how it kind of folds inward? And that's where that area is. So when you look at the brain, I'm going to show you in a second. When you look at the brain, in order to see that primary olfactory area, you have to pull the lobe, some of the cerebral matter back from the lobe, from the temporal lobe, because that olfactory area or cortex is kind of on the underside. You know, if you go back to this picture here, that you have to pull this back to see it. So notice that in this picture, the pink area is the olfactory cortex, and it kind of partially covers up this gustatory or taste cortex right here. So that, you know, you have to pull it back with this forceps here to see that. So that's where the primary olfactory area of the cerebral cortex is. And this is, of course, let me go back to this picture. Notice that it's it's on the same side of the brain. It doesn't cross over. So what you smell with this nostril or this side of your nose is going to that primary olfactory area. What you smell here is going to that primary olfactory area. not crossing over or decussation. What they don't show in that picture is that some of this information, these axons, there's also axon collaterals that lead to the limbic system. The limbic system is the part of your brain that deals with emotion, and it often ties emotion to certain senses and to the autonomic system. And in fact, it's because the axons transmit impulses to the limbic system that your sense of smell can elicit the most strong emotional responses or strong memories, because the limbic system is also involved with memory. And so you might notice that you smell something and it brings back a memory, like you smell fried chicken and it may make you think of your mom frying chicken, or you smell some perfume and it makes you think of a person who wore that perfume, or cologne that makes you think of a guy that wore that cologne, or some other experience. Oftentimes you'll notice that smell elicits a really strong memory because your smell information is going to the limbic system directly, and other senses are not so tied to the limbic system as smell is. Hypothalamus of your brain is also connected. So hypothalamus, which is involved in the autonomic nervous system, is involved in connecting the sense of smell, in this case, to visceral responses. So emotions and memory, that's the limbic system. Visceral responses, that's the hypothalamus. You might notice you smell something doesn't smell good, and it makes you feel kind of sick or gives you a yuck feeling. If you're smelling poop or something like that, or somebody vomited, that smell brings on a gut response. That's because your smell information is going to the hypothalamus, and the hypothalamus elicits that gut response of being maybe nauseous or feeling sick when you smell something bad. There's also an area in the brain called Area 11. I'll show you where that is. It's in the frontal lobe, and that's where odor identification and memory is stored. So you remember an odor specifically. It's kind of an association area. So again, temporal lobe. There's that deep olfactory. It's not deep, but it's kind of, you have to pull it back to see it. The olfactory cortex or area. So they don't show it here because the olfactory area is under. Like, find the gustatory area. The gustatory area kind of, it would be actually underneath here. You'd have to pull this back. Under that gustatory area, you have to pull this back where you have the auditory area. Pull that back, and underneath that green area is where you'd find the olfactory area. Area 11, this is an area that allows you to recognize what you smell. So you smell a flower, perfume, or whatever, onions. It allows you to recognize odors. So that's more of a frontal lobe thing. So that's kind of an association area. Area 11 does some other stuff too, but that's one of the things it does is odor identification. Any questions about that? Okay. As it says here, there's a low threshold for stimulation. It only takes a few molecules of a particular chemical to elicit a sense of smell, or to allow you to perceive a sense of smell. Again, though, we have a smaller area, so we are still less sensitive to smell than other animals. And then with that, also you might notice that you have a fast adaptation to odor. You smell something, initially it smells really strong, and after a while you just don't even notice the smell anymore. So you work in a restaurant, you might not notice that your clothes smell like fried chicken or whatever food you're serving. Or somebody smokes, and they walk in the room and they smoke, but they have no idea that their clothes reek of cigarette smoke. So your sense of smell adapts very quickly, so that you smell it at first, but after a while you don't smell it so much. And I think that's one reason why somebody who cooks a meal, they don't have as much of an appetite to eat the meal after it's been cooked, because they've been smelling it and they've lost that, sense of smell of it, versus somebody who walks in the kitchen, they smell it, they're hungry, they want to eat that food, but the cook has been cooking it and they've lost that, they don't smell it strongly, so they've kind of lost that interest in that food. Any questions about olfaction before we go on to gustatory? Yes?
Does your olfactory nerves affect your taste?
Mm-hmm. As a matter of fact, it does. That's one of the things they talk about is the sense of taste is less sensitive. There's fewer things that it responds to. It doesn't have like 10,000 different things it responds to, it only has a few things. And so a lot of taste is tied into, or a lot of your taste is tied into your smell. And that's why if you have a cold and you can't smell very well, things don't taste as good because of that. So that is true. So smell or taste is really a combination of smell and taste. Now the taste buds, we often think of taste buds as those bumps on your tongue. Those are actually called papillae, and we're going to see what they, see them in more detail. But they contain the taste buds, but the taste buds are actually down inside grooves between these little bumps, between the papillae bumps. And some of the papillae don't even have taste buds in them. Some of them are just there for friction. The taste buds are where the gustatory receptor cells are. And gustatory receptor cells are actual receptor cells as opposed to a first order sensory neuron. They stimulate a first order sensory neuron. In the taste bud, you have three kinds of cells. You have supporting and basal cells, like you do in the nose. And then you have the gustatory receptor cells. The basal cells are stem cells, just like in the nose. They divide and become supporting cells, which eventually become receptor cells. And notice that receptor cells only live for 10 days. Your sensory receptor cells for taste don't even last as long as your smell receptors. They last only for about 10 days before they die, and they have to be replaced. So again, you're getting replacement by these basal cells, which are stem cells. And remember that stem cells are cells that are able to divide and become a specialized type of cell like this receptor cell. Now your taste buds are not just on your tongue. You've also got them on the soft palate there at the back of the roof of your mouth. You have them in your throat or pharynx, and you also have them on the epiglottis, which is a little lid that closes over your larynx so you don't, suck food down the wrong pipe. You have this little epiglottis down there. And so you actually have taste buds down there, though you may not really notice that taste. It feels like it's all in your mouth, but you do get some taste sensation there. There's about 50 receptor cells per taste bud, called gustatory receptor cells, gustatory meaning taste. And each one has a long cilium, or gustatory hair, or microvillus, I guess is what it is, but basically it's a long hair called a microvillus that projects through the taste pore. So here is embedded in the stratified squamous epithelium on the surface of your tongue. That's what you've got on the surface of your tongue, the stratified squamous epithelium, and connective tissue underneath that. That's the outer surface where the saliva would be. You have these taste pores, and where they open are these, what they call microvilli, or gustatory hairs. There's one hair per cell. The cells in blue are the gustatory receptor cells. Now these cells, when they are stimulated, they release neurotransmitter that stimulates the dendrites of first order taste neurons, which are sensory neurons. So these are the actual receptor cells that stimulate the first order sensory neurons, as opposed to the olfactory receptor cells, which are first order sensory neurons. You can also see supporting cells. They don't show, I don't know, there's some basal cells in there too, as well. This is what this actually looks like under the microscope. Here, this is a taste bud. This is a taste bud. This is a taste bud. Here's the little taste pore where the gustatory hairs are. So this cell has been, or this taste bud has been enlarged. You can see the nuclei of those cells in there. And that's the taste pore. You can just barely make out something there, which would be the gustatory hairs. And then down here below would be the connective tissue, where at this level is where the sensory neuron is, where the first order sensory neuron is. Now the taste buds themselves, we're going to look at very briefly, there's different kinds of taste buds. The ones that are called valate or circumvallate is another name. These are in the back of your tongue. There's about 12 of them. They're rather large taste buds. And each one contains 100 to 300 taste buds, so they contain the most taste buds, and they're at the very back of your tongue. And by the way, these bumps are called papillae. These are not taste buds, but they contain taste buds. So Valate is the most common, and it's or not the most common, but it's the one with the most taste buds, and it's in the back of your tongue. Fungiform, as in fungus. They're mushroom shaped, hence the name fungiform, which means shaped like a fungus, or in this case, shaped like a mushroom. They're mushroom shaped, kind of flat on top. They're scattered all over your tongue. They contain about 5 taste buds each. Foliate, these are along the sides, the lateral sides. They are found in children, but they degenerate as children get older. This is probably why children have a stronger sense of taste and why they don't like certain foods very much, because it tastes very strong to them. But as you age, those taste buds go away and your taste sensation gets less. So part of it is just children have to be exposed to certain foods. They say that it might take 15 times before a child will accept a certain taste that they have to learn to like. Maybe they won't learn to like it, but it takes several times before they learn to like something. But part of it too is, I remember my roommate in college saying that when she was a kid, vegetables tasted really green to her. They just tasted really strong. And that was probably what it was she had those extra taste buds that we don't have as adults. Phyloform are thread-like. Phyloform means thread-shaped or thread-like. These are all over your tongue as well. These do not have taste buds. They provide friction on your tongue so that you can move food around in your mouth and swallow. If your tongue were perfectly smooth, or if it just had these taste buds, it would be harder to swallow and hard to move food around your tongue. But if you look at a cat's tongue, they have those, almost sharp taste, or papillae, not taste buds, but papillae. Those are phyloform ones that are more elongated in order to help them clean their fur. So these are elongated thread-like ones. And so here's a picture. This is the tongue this is the front of your tongue all the way down into your throat. The palatine tonsils are those tonsils behind your soft palate. If you still have those tonsils, you can see them peeking out behind the soft palate. The epiglottis is way down here in your throat. It's what covers the larynx when you swallow, so you don't get food down the wrong pipe. There are taste buds back here, including on the epiglottis itself. They're sticking up, but when you swallow, it comes down, it forms a lid over your larynx. Back here, these big bumps are the valate or circumvallate papillae, and then you've got the fungiform and phyloform ones all over your tongue. Those foliate ones from childhood will be over here in the margins. And so here's a picture that illustrates the different shapes. Circumvallate's the biggest and has the most taste buds. Phyloform, or the fungiform, is kind of broad on top like a mushroom, and it has fewer taste buds than the valate does. Phyloform is thread-like, kind of sticks up, pointy, kind of pointy looking. And then here's a picture from your textbook showing the same thing. So here's the big circumvallate or valate, there's another name for it, lots of taste buds. Those are phyloform ones that give friction. And then here's a fungiform that's supposed to be shaped like a mushroom. Though valate and fungiform almost look the same, but the valates are actually bigger and have more taste buds in them. And then here's an electron microscope image, 60 times a magnification of the surface of the tongue. And you can see the blue ones are phyloform, they're more narrow, and then the big red ones are fungiform. These are scattered all over the tongue surface, whereas the valate ones are in the back toward the throat. Any questions about that so far? Okay, we've just got a few more minutes, so we'll try to get through as much as we can. There are 5 types of taste, but there's been a lot of research on taste receptors, and they're discovering new receptors, as they go along. But there's what are called sour, sweet, bitter, salty, and umami, which is savory, like when you eat a steak or something with a lot of protein. Savory is basically something with a lot of protein in it, so if you're eating a steak and things like that. And as we were talking about, the combination of smell and taste creates the other flavors, because we have more limited taste in our tongue and in our throat, but when you combine that with the smell of something, that elicits more of the flavor, which would be like if you're eating chocolate or spices like cinnamon and coffee and chocolate, things like that. Olfactory receptors are more sensitive than taste receptors, so it takes more to stimulate those taste receptors as well. Now, the threshold for taste varies. Bitter tastants or chemicals, tastants are the chemicals that stimulate a taste. Bitter ones have the lowest threshold of stimulation, and this is probably a protective mechanism because many toxins, especially plant toxins, are bitter, and this protects you from eating something poisonous. However, a lot of the foods we eat are bitter, but we've been, selectively breeding plants so that we could eat these bitter compounds and not be poisoned by them. So, but bitter is often what plants do. They make bitter compounds so that animals won't eat those things. So that's just a protective mechanism. So very quickly, hold on, we got a couple minutes. This is the gustatory receptor cell right here. This cell, when it's stimulated, it's going to generate a receptor potential instead of a generator potential, because that receptor potential is what stimulates the release of neurotransmitter. That neurotransmitter binds to the dendrites of that sensory neuron, and that sensory neuron is unipolar, and that generates an action potential that goes to the brain and to the eventually to the gustatory area. And what this picture shows these are different taste receptors that are stimulated by salty, sour, sweet, and umami, and bitter. And there's different ways they are stimulated. They all release neurotransmitter that stimulates that sensory neuron, that first order sensory neuron that carries information to the brain. So on Tuesday, we'll talk about how they are stimulated, and we'll finish up with taste and get into the eyes, and we'll cover all the eye stuff on